right, I wanted to put together a little presentation so that we could see meiosis in kind of an animated setup. So let's start talking about meiosis. So in our first slide, we basically are talking about an organism that has 6 chromosomes. It got 3 of those chromosomes from its mom. Okay, so we can see those 3 chromosomes in pink. And it got the same type of chromosomes from dad when this particular organism was formed. So that's why I have them in kind of the pinks and the blues. So you get the idea that this organism was also formed from the union of an egg and a sperm, and got half of its chromosomes from its mom and half of the chromosomes from the dad. Okay. Now this organism wants to produce gametes. It wants to produce either egg or sperm. And so it's going to have to go through the process of meiosis. In the first step of meiosis, in prophase 1, each of the chromosomes in that cell is going to duplicate itself. And when it duplicates itself, it's going to form 2 sister chromatids. Those sister chromatids are united by a centromere, so we can see this chromosome is duplicated, the blue from number 2 dad, and it's joined together. So each of those 6 chromosomes has made a copy of itself, getting ready for this process of meiosis. Okay, once those chromosomes have made copies, then in metaphase 1, remember that metaphase starts with M, that means they're going to line up in the middle of the cell, but in meiosis, they line up in pairs. They line up in those homologous pairs. Homologous pairs means that I have each of my number 1 chromosomes lined up next to each other, each of my number 2 chromosomes lined up next to each other, and each of my number 3 chromosomes lined up next to each other. Okay? When they're in this metaphase 1 position, I have 4 chromatids. I have a chromatid here, a chromatid here, a chromatid here, a chromatid here. I've got all 4 chromatids together, and we call those groups tetrads. Okay, now it doesn't matter. I've lined them up in one way, but they could just as easily have lined up like this. Okay, they could have lined up like this, or they could have lined up like this, and I didn't draw all the possibilities. This is the concept of independent assortment. Each pair of homologous chromosomes lines up on either side of that midline independently of every other pair of chromosomes. Okay? That independent lining up is going to create a lot of variation in the gametes that can be formed. Okay? We're going to go with this particular lineup and we're going to move forward. The next thing that comes after metaphase 1 is anaphase 1 and telophase 1. And in anaphase 1 and telophase 1, we're separating these homologous pairs of chromosomes. So, and my little purple line down the middle is telling you, hey, this is now divided into 2 separate cells. So each cell still has duplicated chromosomes. I still have 2 sister chromatids for chromosome 1, chromosome 2, chromosome 3, but my homologous pairs have separated from each other. Once that has happened, these chromosomes in each of these 2 cells is going to line up in the middle again. This is going to be metaphase 2. So again, my line down the middle is telling you we're looking at 2 cells here. Now my sister chromatids are going to separate. And I've kind of already drawn, we're starting into anaphase 2 because I've got those sister chromatids. They're not connected anymore. They're starting to move apart to opposite ends. So we're looking at anaphase 2 and telophase 2. At this point, those sister chromatids are going to go to opposite ends of the cell, and now I have 4 haploid cells or haploid gametes as a result of meiosis from that one original diploid cell. Okay, so each cell has only 3 chromosomes in it, whereas my original cell had 6. And they've got different arrangements of the chromosomes depending on how those homologous pairs lined up. Okay, so this is just, again, kind of an animated version of meiosis to hopefully make it make a little bit more sense for you.